Well, it is very important to address actually COVID-19 and psoriasis. And in many countries, we are suffering on the challenge of COVID-19, and especially also in countries in Latin America, I realize that this is a big problem. And we are dermatologists, and we are taking care of our patients with psoriasis in a very difficult time. The first question which I would like to address is the following. Are patients with psoriasis at risk, at an increased risk in a pandemic of COVID-19? And I would like to address your attention to an important study which was on the association between COVID-19 and skin conditions. The study, one of the principal investigators is Janssen, Johan Janssen, also an IPC board member. And they investigated a patient collective of 435,000 patients in the Michigan area. And they looked at skin conditions. And what they actually found out was that patients with psoriasis and atopic dermatitis, and this is in contrast to what I expected, had an increased risk to get COVID-19 and to get symptoms of COVID-19. But, and then the good news is they had a decreased risk of mechanical ventilation. And this is the entire population of patients with psoriasis. These associations, so, were not due to the immunosuppressive treatments. And what they did is they carried out also gene expression studies and showed an overlap in gene expression between infected normal bronchial epithelial cells and inflammatory cells in psoriasis and atopic dermatitis. For S100 family members in the epidermal differentiation complex was shared. The IL-17 signaling pathway was shared. And also in a genome-wide scan, they also identified indeed a locus for S100A12. And what they concluded is that there are shared components involved in anti-COVID-19 immune responses. And let's move now to the pathomechanism of COVID-19. SARS-CoV-2 is entering the body and is binding to the ACE2 receptor on lung epithelial cells and gut epithelium. And then an immune response follows. And in this immune response, actually, there are two parts. One part is the viral clearance, which is the acute phase of inflammation induced by COVID-19. And there is the more long-term response, and that is hyperinflammation. And what we see in the viral clearance phase, that cytokines important in atopic dermatitis and in psoriasis, that these cytokines are also crucial in the early phase of COVID-19 infection, which is dedicated to viral clearance. IL-15, interferon alpha, beta, gamma, IL-12, and IL-21. And it is attractive to speculate that in patients with psoriasis, the machinery is there already of these cytokines to over-response. And that patients with subclinical signs and symptoms of COVID-19, just in patients with psoriasis, may have an increased expression of inflammation and be recognized in epidemiological studies. But what about the chronic phase? If patients really, by a series of still unknown mechanisms, go to the phase of hyperinflammation, then also we see that important cytokines are very important in this chronic phase, like TNF, like IL-17. And it is indeed attractive to speculate that in the long-term phase, it is important to inhibit these cytokines. So early inflammation with increased expression of IL-17, IL-12, and IL-21 may be protective, whereas in the longer phase, indeed, we need to inhibit these enzymes. But now let's approach the different targetings of the various inflammatory diseases. Then we know that anti-TNFs are very important in colitis ulcerosa and Crohn's disease, and in psoriasis and spondyloarthritis. And we know that indeed IL-4 and IL-13 interference is important in atopic dermatitis. And we then ask the question, to what extent is it permitted from safety point of view to target these pathways? When is it that we get a green light with respect, for example, to targeting TNF-alpha? When we consider viral infections, we get a green light. There is no problem, but we know that we get a red light for anti-TNF treatments for bacterial infections. And we know this also from tuberculosis, that patients on anti-TNFs have an increased susceptibility to get active tuberculosis or to aggravate tuberculosis. Fungal infection, also a red sign. And when we look to the IL-17s, we see that viral problems are not the issue, but that fungal problems are the issue. But when we follow this line, we see that across the currently available biologics, there is green light for the various biologics. An exception is JAK1, JAK3 inhibitors, where we again have a red light for viral infections. Hyperinflammation in COVID-19, the cytokine storm. There is the acute respiratory distress syndrome, where there is massive inflammation. There is the macrophage activating syndrome in virus-induced hemophagocytotic lymphohistiocytosis, and suggesting a significant role of the host innate immune system in this chronic phase. And we also know that characteristic of this phase in COVID-19 is microthrombi, which have been identified in nearly all major organs. And what about the skin? Also in dermatology, we have described well-known changes in the skin. Coagulopathy and thromboembolic events in patients infected with SARS-CoV-2 infections long-term are well known. We know about the well-known cardiovascular risk factors, and in the pathogenesis of the thromboembolic complications, we know that an unrestricted angiotensin II action is of major importance, resulting here in endothelial activation, complement stimulation, and an activation of the innate immune system. And when we now move to the skin, we see from this Spanish study that several skin conditions are seen more frequently in the long-term phase of COVID-19. The so-called pseudochilblain, vesical eruptions, and indeed often also with hemorrhagic content, urticarial lesions, maculopapules, and livedo or necrosis. Please visit the IPC website, www.psoriasiscouncil.org, and there is a research page on COVID-19 and psoriasis, which contains national guidelines, provides videos of experts, and lists of important publications. The IPC view is more evidence is needed from real-world practice before firm conclusions can be drawn on the safety of immunosuppressive treatments. And therefore, we support very much the PROTECT registry. With respect to recommendations and statements, indeed the IPC was one of the earliest international organizations with a statement. And this statement was indeed that when patients have very have active COVID-19 infection, in that situation, we better postpone a treatment with a systemic anti-inflammatory drug. But when patients are actually have no active signs of symptoms of COVID-19 disease, in that situation, at first we said we have to look it on a case-to-case based situation. For there are additional risk factors which are also comorbidities of psoriasis. But it's very important to build our strategy on best evidence. And there is the PSO-PROTECT study, and the first publication out of this PSO-PROTECT study revealed important new information. In this international case series of patients with moderate to severe psoriasis, biologic use was associated with a lower risk of COVID-19 related hospitalizations than with the use of non-biologic systemics. However, further investigations warranted on account of potential selection bias was set, and established risk factors were indeed reconfirmed in this study. But now we have more than 1,000 cases in the PSO-PROTECT registry, and we see from the various countries of the world indeed that there are cases accumulated. And also US dermatologists can contact psoprotect.org, and it is well appreciated that you report your patients with COVID-19 and psoriasis. But when you look to the analysis, we see that on the question, was the biologic medication for psoriasis stopped during active COVID-19 infection, there was a yes in 45% and a no in 52%. And when we look then to the outcome of COVID, we see that the vast majority, 93% of the patients showed indeed recovery. And we move indeed in our understanding of for the recommendation. Evidence is accumulating from real world evidence studies regarding immunosuppressive treatment in the context of COVID-19. And if the patient is on biologics and there's no COVID infection, biologics can be continued and patients are to some extent protected for severe COVID. Non-biological systemics also can be continued if the patient has no active COVID infection. But in case of active COVID-19 disease, then discontinuation of anti-psoriatic treatments still is the practice by most dermatologists. But we have to realize that now in PSO-PROTECT, half of the patients did not continue biologic medication during active COVID-19, and 93% showed recovery. During COVID-19, we have learned to use more intensively telemedicine. And there is a study in the United States where patients were asked about the value and the acceptability of the use of telemedicine. And it was very positive, the outcome. Time efficiency was by 81% indicated as a major advantage, not requiring transportation, maintaining the social distancing. Lack of physical touch was indicated by 26% of the patients. And some patients felt that they had not the most adequate assessment. But very few patients reported that they were unlikely to accept a further telemedicine guidance. When we look to psoriasis and telemedicine, we have seen that it's in a study by April Armstrong in an online model that this enables patients and primary care physicians to access dermatologists online asynchronously, which is well appreciated. The dermatologist provided assessments, recommendations, and education and prescriptions via the pictures, the store and forward approach, and via web page interference. And then they evaluated the efficacy in the group of patients being treated in the classical way and with telemedicine. And what to my surprise, they found that those patients who were guided by telemedicine even had a more favorable outcome of the improvement of psoriasis in objective severity assessments. We will learn more by the telemedicine working group of the IPC on the possibilities and the added value of telemedicine in the guidance of patients with psoriasis. And now let's move to vaccination. Vaccination, I think there has been done a tremendous development, both in our understanding of the vaccination process as the development of vaccination platforms. The classical vaccination platforms are the so-called inactivated virus vaccine and live attenuated vaccines. This is a time-intensive and often a very long-term development process to develop specific vaccines. New approach are recombinant protein vaccines based on spike proteins, and here recombinant techniques on the receptor binding domains. And this approach is a more selective approach, but the problem is that the immunity induced in this way is the humoral immunity and not the cellular immunity. Recombinant protein vaccines based on virus cell particles also is part of this. And then very important, that are the so-called vector vaccines. We have replication incompetent, replication competent vaccines, and inactivated vector viruses. And in these vector viruses, actually a small piece of DNA is incorporated, often targeted at the spike gene of COVID-19. And then we have the DNA viruses. I will be very short, for there are new viruses in development but not registered. And there are the RNA viruses, which is truly an innovation in vaccine development, which came really at the right time in the COVID pandemic. I will guide you through the different approaches. That is, let's have a look at the platform RNA virus. We have the Moderna and of course the Pfizer vaccine, which is RNA technology. Important is that the immunity induced is both humoral and cellular. And we make use of the genetic sequences, and the great good of this approach is that quite easily the vaccine can be adopted. We can follow the mutations. We can follow the virus mutations by a quite fast process in developing new vaccines when this is needed. Then there are the non-replicating viral vaccines. There's the AstraZeneca vaccine, and not on this slide, but also very important, the Janssen vaccine. That is another virus which is used as a vehicle. And in this virus, then DNA against spike protein is incorporated. Very important, humoral and cellular activity, immunity is induced, can be manufactured on large scale, and I think that's also very important. There is the classical approach with an inactivated virus, and this takes indeed quite a long time to develop this virus, but the downside, it's most to humoral, the immune response which is induced. There are sometimes reported some adverse events of vaccinations, and very well known to all of us is the interim statement on the COVID-19 committee of the WHO Advisory Committee on Vaccine Safety. That's the AstraZeneca COVID-19 vaccine. And what has been found is indeed thromboembolic complications. These thromboembolic complications also occur in patients younger than 60 years, and several of the patients, indeed, the fair number of patients are women using oral contraceptives. And the committee feels that a causal relationship is plausible between the vaccines and these complications. But we have to realize that 200 million individuals received the AstraZeneca vaccine. Individuals who experience any severe symptoms is the advice, such as shortness of breath, chest pain, leg swelling, persistent abdominal pain, neurological symptoms such as severe persistent headaches, blurred vision, tiny blood spots under the skin beyond the site of the injection, and from around 4 to 20 days following vaccination should be checked with thromboembolic complications. So the WHO pays attention to this, saying it's a very rare event, but let's be aware of the possibilities of thromboembolic complication in this respect. What this slide shows a little bit is the frequency. That is, the AstraZeneca vaccine is 4 cases in 1 million vaccines. But birth control, we have 500 till 1,200 cases in 1 million women using contraceptives. And smoking, 1,700. And COVID infection itself, 165,000 cases in 1 million. So numerically, we will see that there are very seldom side effects reported with the vaccines, but this is not immediately a reason to say that the vaccination with these products should be stopped. We have to look here at the epidemiology. What type of immunogenic response are elicited by the SARS-CoV-2 vaccines? Very important is that the neutralizing antibodies to induce that in itself is not sufficient for adequate vaccination. But with the inactivated vaccines, we induce indeed mainly neutralizing antibodies. The live attenuated vaccines, luckily for us in psoriasis, there is not yet available a live attenuated vaccine for the vaccination of COVID-19, as we know very well, is that this vaccine is contraindicated in patients using immunosuppressive treatments and using biologics. Viral vector vaccines can be given as these viral vector vaccines belong to the non-replicated form. Very important, humoral response and a T-cell response. The subunit vaccines, that's the protein vaccines, again only induce humoral immunity and are likely to be less active. And nucleic acid vaccines, RNA vaccines in particular, I think this is a major innovation which really is so important that we have this available. What is the International Psoriasis Council making as a statement on after that the vaccines program now has started? Firstly, avoid live attenuated vaccines. Luckily, not yet available they are, but they may come. And please realize that immunosuppressive treatments is really that prohibits the use of the live attenuated vaccines. Currently, 3 vaccines are used, and these are not live attenuated vaccines, as we know. That's the RNA-based vaccines and the replication deficient vector vaccines. We anticipate that most patients with psoriasis who do not have contraindications or known allergy to vaccine will be recommended to receive one of these vaccines soon on local availability and guidance from the local public health bodies. And what we advise is not to be too complicated on this. It is important that patients with psoriasis and also patients using biologics and using the systemic immunosuppressive drug get vaccinated. And when you start by saying, no, you only can be vaccinated if you have discontinued your biologic for at least 3 times the half-life, or you have to discontinue your methotrexate, actually the vaccination program is disrupted in these patients. And let's go for vaccination. Let's make it simple. The effective vaccines in this population will need then to be established in registries where we have got to build up our evidence also in patients with psoriasis and with the various drugs, how they will respond. But let's do that by learning. Important is that we get vaccinated. There's no evidence that vaccine affects psoriasis onset and severity, and it's important that all people with psoriasis have access to adequate care, including the access to SARS-CoV-2 vaccines. So, in conclusion, I think what is building up is the evidence. Firstly, we know that patients who have psoriasis in the pandemic of COVID-19, that you know that patients on biologics have not an increased risk, they have a decreased risk of indeed getting severe COVID-19. Patients with active infection with COVID-19, there is the evidence building up that maybe the continuation of the COVID-19 is not so harmful for the patients as we thought before. This is also important for many of our biologics have indeed a very long-term elimination half-life and a special immunological duration of the response. And with vaccination, get vaccinated. That's the message to the patient. Thank you very much.
Fantastic, Peter. Thank you for such comprehensive topic. And you get from immunopathological view until the most practical view of what we should do or not do. And unfortunately, sometimes we will have to wait for more answers. So thank you very much.